Johan's second lecture. Take it.
Okay. Okay. So yesterday I talked more about, in general, the theories that of how species interact. It was basically not my own work. Today I'm going to change that. I'm going to talk much more about my own work and my own adventures in spatial ecology, as you could say. And so it's a bit of history, and it will end up with this. But although I will present this beautiful ecosystem only on the last day. But of course, this is a marvelous picture, isn't it? I mean, look at this photograph. It's a very large, for Dutch standards, salt marsh in the south of the Netherlands, and you see all the patterning there. I mean, you see the sand dunes in the middle, but all the creeks and the vegetation aligning along the creeks. And you might think like this: well, this must be hugely complex. All different processes that are shaping this landscape. Or is it actually, maybe, are there still simple underlying processes that shape this ecosystem? And that's basically what I want to talk more about in the coming days, actually. And I will actually start simple. I start with basically with the basics. So today is about spatial pattern formation well, as an example, arid ecosystem. I will start with that. But we'll also talk about mussel beds and also back to some sort of generality. I like historic context like I did yesterday, but I will also work from my own experiences today and basically tell how these ideas about pattern formation in ecosystems come about. Okay, so where were we yesterday? We were here. This is the predator-prey model that you saw yesterday. And of course, this is a simple model that presumes that you have an ecosystem that's an homogeneous, well-mixed thing where interactions are equally strong everywhere in your domain. And the domain can be like a little pool on a rocky shore or something, like maybe just a meter. Or maybe it can be a massive ecosystem like 5 by 5, 25 square kilometers, like the one I showed you in the previous picture. It isn't defined. But of course, if you if you have ecosystems like this nice picture, the shark travels between Florida and Greenland on that scale. That's the scale at which it moves, and it's basically a very migratory species. While the little fish there, they probably don't go any further in the coral reef at which they are living, and they really rely on the protection of the coral reef, so they're much less mobile. They don't operate at the same spatial scale. And that's an important thing to pick up. There are these scale differences. Okay, so yeah, is space important? Could we explicitly include space in our models? Of course, that's I already seen by the previous lecture that you can do that, but given an ecological context, that would be an interesting question. Would interactions change? Would it be more stable, less stable, whatever? Could ecological interactions, are they depending on spatial heterogeneity, or can they actually even create spatial heterogeneity? Can ecosystems be patterned because of the interactions? And these are, of course, important questions that to work on. And, well, spatial interactions. What processes make ecological interactions spatial? Who has an idea about that? Give me some thoughts about a spatial process that you would want to include in the previous model. Anybody? Yes. Movement of animals. Animal movement. That's a particular sort of movement, of course. Animals are, well, they go about, they are searching for food, or they just have eaten the grass here and think, I will move a little further on. So they move to avoid depletion. There's all sorts of reasons for movement. You can move it because you're migrating. But there's also, well, other examples. I'll let you, yes? The resource, well, it's about movement, of course, types of movement.
No, I mean like the actual food could be like a gradient of food, or it could be that only around like the delta of a river or stuff like that.
So what would actually move then?
No.
Well, you have a reason. Now you are giving a reason for movement. that there's heterogeneous conditions and you want to move to one condition to another. It would be still be an animal. But for instance, one other movement could be the dispersal of plants. Plants disperse mostly passively, sometimes actively by hitchhiking on a bird. And so that is a form of dispersal, which could be, as you say, because of different conditions, and you want to disperse yourself to a new condition. Well, I made I made a little list. So animals move actively, which is the most well-known form of movement, of course. Plants disperse, but animals migrate. There you have these famous migrations on the Serengeti. It's not that they are at that moment searching. They just know, I want to go to the other side of Serengeti, and they just go there. And then when they arrive, they start searching again. And so basically that's just migration for a purpose. But you can also just move along with the water. Maybe you're a little bit phytoplanktic species, and you're in the water, and the water moves through the ocean. So you're not actively moving yourself, your substrate moves. And of course there's diffusion. The plankton is for a part diffusing. So there's physical processes that drive that. So all sorts of forms of movement. And so how do you then do that, put that into a model like here? Well, of course, we know a couple of the terms that we use for that. We have diffusion as a way to include movement in a model, some sort of model, a population model with a logistic growth, for instance, or advection, when there's a force that drives organisms away. But of course, diffusion is a process of, well, can you say that animals are moving in the same way that they are just moving about? Are they just then diffusing? That, of course, is always a bit of a tricky question, because animals are just a little bit more complicated than the atoms and so on. And so, well, I want to go back to the basics of that, where it started. I think it will be a bit of a repetition for you guys, but this is how I start with an ecological lecture, is that, well, the basics behind how we actually model the movement of organisms in ecology is the same way it is done in many places. We assume that Brownian motion occurs, random movement of organisms. And Brown, I guess Brown was actually a sort of a botanist, ecologist, as you would say. He was not at all interested in Brownian motion. He would preferably have not Brownian motion. He was interested in pollen and how, if you would measure in a lake, in the sediment of a lake, if you could see how the vegetation in the lake or surrounding the lake would have changed in time during millennia, and he would put them under a microscope and try to identify them. But yeah, the whole stuff was also constantly moving and shaking. I couldn't see what it was because it was shaking. And so he was intrigued by the shaking. He actually started cooking them and see, like, well, I just killed them, then I can see where they are. Didn't help. They were still moving. And then I put clay in the water, and the clay started moving. And so on the scale which you see here. So he basically understood that the movement, Brown, that the movement was a physical process and not necessarily active movement by the organisms. But he never solved that. And, do you guys know who solved that? You guys don't know who solved it, then who else will? So who solved that? Einstein, indeed. And so this is for ecologists. This is what? Einstein? And so, yeah, Einstein solved the problem. He basically derived that the random movement of atoms was his proposition, and in the time, the random movement of particles, you can build from that how ink in water diffuses through a substrate. One of his very famous papers. I guess you guys know more about it than I do. What I always mention is that people always have this picture of Einstein as a very old guy with gray hair, standing upright and so on. But that was not the Einstein who did that work. This was the Einstein who did that work. I was about 26 years old when he published it, 25, 24 when he did the work, I think. How old are you guys? Suggestion. Don't wait until you get gray hair before you want to make a breakthrough, because I can assure you it's too late. So, anyway, go on. This was picked up decades later by people modeling populations, and this Fisher is a very well-known name in population dynamics and statistics. Fisher exact test, all these sorts of things. So a very, very famous population biologist, and he just took the logistic growth equation and put the diffusion term in there, and just see how it could explain the spread of a gene in a population. He was very much into gene flow and genetic processes. Actually, if you want to read something very interesting, you should look at the Wikipedia page of Fisher, and you see all sorts of things about eugenetics and racial differences and all these sorts of things. Very interesting. Very, well, strange, as you can say. But it's still, this equation was one of the first instances where an ecologist used the diffusion equation to model population spread. And so now I want to make a bit of a leap. I want to work on basically the model. I should have put in a little equation here. So this was the model that we had. It's a slightly different form now, but it's the model with a logistic growth equation, as you can see here, and holding to a functional response, and so on. And so if you have this model, what you can, of course, do is add to this predator-prey model the dispersal terms and see how this works. Well, this was done by Mike Hassel, who was, well, basically interested in these things, and he tried that and put this in and made a very nice publication out of it. And so basically, I followed the model of Girard. You add here the diffusion terms with using a gradient operator to describe diffusion, and this time in 2 dimensions. And well, what do we ecologists do if you make a model like this? You see that people with a good physical background start to draw all sorts of complicated graphs and equations, but we just put it into a computer and just press run and see what happens. And that can be really interesting. Oh yeah, I have some explanation here. So you can see this prey dispersal and predator dispersal added to this model. So then, if you run a simulation, something really interesting starts to occur. I guess you guys have seen something similar already. But in the context of a predator-prey model, it works like this. So it's a predator-prey model, like the one we had yesterday. So it has a tendency to oscillate. It goes up and down and up and down. But as time progresses, strange things happen, as you see in this model. Patterns start to develop, very dynamic patterns. Isn't it cool? Of course, these models are found in various forms. But of course, if you put this into the context of an ecosystem, it has really some important consequences. And you can see that, well, let's run the model again and see. So what is if you don't look at all these nice patterns and the colors and the flashy effects and so on, but really think like, okay, this is population. And population changes in time. What happens to this population? So now it's like flashy, flashy, flashy. As time goes on. I don't remember anymore, but I guess so, yeah. I guess so, yeah. Yeah. A bit of an old movie for me. So, you can see that they're linked. Yeah, yeah, sure. Periodic boundary conditions, yeah. Yeah. But what's the remarkable effect what you see here? Look at the lectures that we had yesterday, where we found that if you have a highly productive ecosystem, the whole ecosystem starts to oscillate. The predator-prey interaction starts to oscillate. So what do we see here developing? Who wants to make a claim? You were the first.
I'm just guessing, but it looks as if the populations stabilize if you integrate over space.
Exactly.
Yeah.
So, of course, it doesn't stabilize on a square meter scale. A square meter scale is just as dynamic as always. There's another finger pointing up. Would be interested to hear some thoughts.
What I noticed is that there are some places that doesn't have any density, and then it moves and it gains the density. But some places are the place where is the prey is also the predator there, so they move along.
Yes, they chase each other basically. But important is that you have some places that are bare and some places that have still depopulation. But if you look at the scale of the whole thing, there's a certain amount of yellow, and it doesn't flash anymore. There's at the beginning it was looked like a stroboscope, but now it has all been cancelled out. But on the scale of the whole picture, not on the scale of the square meter. And that's an interesting phenomenon. So you have this dynamic system. It creates these chasing waves of prey and predator, but remarkably, because of that small-scale dynamics, it stabilizes on a large scale, as you can see if you plot it. And this was considered by Mike Hassel as something really important, because you can see here that the initial simulations with these oscillations, it really crashes to zero. And of course, in this model, you can have a population density of 0.000001 individuals. But yeah, I mean, that doesn't work in the real world. So that means population extinct. But because of these patterns developing in this intrinsically unstable ecosystem, you see this, it all stabilizes. And so that was actually a sort of an emergent property, as they phrased it, of the pattern formation that occurs within this system. So that was a remarkable result, and that's also the reason why it ended up in a nice journal, in Nature. And so that was a remarkable insight. Of course, as always, you have the modelers and you have the field ecologists. I always like to make that comparison. So what do you think that what the real rubber boot ecologists would think about this sort of modeling stuff? Sorry, I didn't see. Sorry.
So I'm also imagining that the dynamics probably has some amount of a lag from previous configurations and basically has some sort of predictive power.
The specific configuration, well, if you start with the same exact configuration, you will, of course, it's a deterministic system, so it just produces the same pattern. But it uses some sort of pattern with a certain property, but the specific location where these patches are is not stable, and it changes constantly. So it's that way, it doesn't really have a history effect if you look at the scale of the whole box, so to say. So in that way, it actually loses at least a part of the history effect. But it gains a sort of a property, a specific dynamics locally with connected in the whole field that sort of stabilizes this ecosystem. Does it answer your question?
Yes.
Okay. Okay. Well, we have seen it now. We don't have to see it again. Okay. So what do you think the field ecologists thought about this? Have you ever gone out for a walk outside in the forest and you were not thinking about mathematics, but you were looking at the flowers and so on? Did you ever see a traveling wave? I didn't. Never seen these. So what do you think the ecologists thought? Like, yeah, this is computer science. This is these guys, they go off in their mathematical world. It has nothing to do with the real world. And, well, I think for this example, it's actually true. There's actually one study that actually shows a little bit, but this is the best example, I would say. Although, I must say, this is the best example for something that works on a real big ecosystem. On a small scale, dynamics, and in petri dishes and these sorts of things. Yeah. Although they typically start with a point, and then they go out, and sometimes they break up, and then you see sort of this spiral things happening a little bit. Yeah. Well, here on this continent, I think in the Andes Mountains, there are vegetations that look also very curvy and have typically, they look very much like this. Although I don't think it's a predator-prey relation, but it does have this, it looks like spiraling. So there are examples where this actually occurs in the outside system. And of course, there are many examples where people have done it within petri dishes with bacteria and so on. That's actually well possible. If you really make an artificial system, you can get this dynamics also in a physical model, so to say. But this is one ecosystem example where you can see there's the vole and the tree sapling interactions, which also sort of shows similar dynamics. They were an example of something that would be showing population explosions. In Finland, sorry, I should explain this. This is Finland. You see the shape in the corner there. And, well, it looks like, well, definitely looks like unstable. You have to do some imagination to see a spiral wave in here. Yeah, and so this led to actually this nice paper, Spatial Self-Organization and Ecology, pretty patterns or anything real? And so there was an argument pointing at actually the lack of real explanations for this phenomena. And this is 1997. It's important to see that this is the time where people hadn't been looking to real-world ecosystems really. And then, of course, this slide doesn't belong here. Where is I messed up my slides. And well, then examples of this sort of systems came up. It's these sorts of examples from arid systems. That typically you find these in faraway places. If you want to see this landscape, you have to go to the Sahel, far away. And you have to go to Sahel and to parts in Africa where there are not too many humans. And people always show these romantic films, movies about Africa with beautiful nature. But the Africa I came where I've been to, has just as many humans as everywhere. So you have to go to really remote places where there's hardly any human effect. And then you see these sorts of bushlands, which are regular patterns. And of course, these aren't spiral waves, but there's a different sort of regular pattern that comes about. And what you actually see these little dots. These little dots are bushes. They are a few meters wide, a few meters high, not really big, not trees, they're smaller than trees. And they are forming gaps in between, as you can see here. And so that's part of the story here. So to go back to my previous slide. So I want to actually start with what was, if you would ask an ecologist in the 1997, what are the notions, what concepts are there about to explain which plant is growing where? I mean, if you want to understand the structure of an ecosystem, what theory is there? He would point you at zonation theory. He would say like, well, you know, there's a landscape, and there's places that are high, and there's places that are low. And you can see in this example picture where it's all spread out in a few meters, you have, well, on a rocky coast, up there it's freshwater. Conditions are like for freshwater plants. As you go down, you have an area where there's lots of wave action, so it's very bare. And you go further down, you go into the intertidal, and then finally into the subtidal regions, and you find a whole zonation of plants along there. And basically the idea is then, well, you have a landscape, and the landscape determines where you find certain species growing. And the idea behind that comes from a classic concept that every biologist learns in his first or second year. It's called the niche theory. Basically, every organism has its own place in nature. A very romantic idea also. And so you have forests, and they grow in places where the trees, they go in live in forest environments. You have the grasslands, then you find grasses. And where you find a plant, is where it grows best. Every plant grows in its own optimal place. Well, that concept was already criticized early on. When, well, if you want to understand mussels, for instance, the little shellfish that you see on the top part there, they're filter feeders. They eat algae in the water. So where to best grow if you're a mussel? Well, in the water. Well, where do you see them? In the intertidal. So where there's actually part of the time, it's emerged out of the water. You can't eat anymore. But the reason for that, I actually think that Bob Payne put the starfish for this photograph along the edge. But they can't grow, they can't live down because they get eaten by starfish. And so the starfish are limiting the survival of the mussels in the lower parts. Of course, this concept has at the back of its mind that the landscape shapes this ecosystem. And so, well, then of course, the question is like, well, is there nothing more than landscapes? Well, there was of course something more, and I already talked to you about the African bushlands, but I want to show you a couple of examples, and you have to figure out what's here. So what you see is the Rocky Mountains in the United States, high up, and you have trees growing there in lines. They're remarkably lines. So what would be causing the lines here? Why are the trees growing in lines? Yes.
Because if they are separate, they can take more
because if the trees are more separate, they can take the solar light for more time.
Or they would better separate,
Yeah.
but they don't.
But they are separated like in some direction. So if the sun, they can take the solar light for one direction from the whole day.
Well, but so you can imagine that this amount of trees would spread out evenly over the whole landscape, and that would then give them less or more light. I would think that they will get more light. So sitting together, of course, the stripes don't influence each other anymore, but within a stripe of trees, you have plenty of competition for light. So I don't think it's a good argument. Anybody else? We have one here.
I'm thinking about wind, maybe.
Getting you to the right direction. But that's not enough. Actually, I will tell you, so where do you think the wind blows from?
I'm thinking like if the wind blows in the direction of the longitudinal direction of the stripes. So
Yes, but it goes exactly the other way. It goes perpendicular to that.
so my theory is completely failed.
No, but I think it's something like that, and I would guess it's about maybe soil and nutrients. You know,
That's a
like
good thing-
it's
good thing to
washing
mention.
away, so it forms little waves. You know.
That's a good thing to mention. That's also what people would argue. But actually, it's very flat, and it's not that the basic underlying soil is any different. Of course, the plants, the trees have their effect, but it's not that there's sort of an independent phenomena in the soil that makes the plants grow there. It's not deeper, it's not wetter, it's just the same as everywhere. So actually, the story is well, one more, of course. Yeah.
The seeds get caught in the plants that are already there, so they grow where the plants already are.
It does happen, but that's not the... Actually, I don't give you enough information to solve this riddle. I will show you later more. Yes?
Am I allowed to guess? It's a little bit sloped, isn't it? A little bit.
I
It
think
seems
so, yeah.
like
I
to
think
the
so,
left,
yeah.
right? Yeah.
Yeah,
So
yeah.
could it be that this, you know, I don't know, if this is a very even slope going for a long way, that you might have some flow of water or some competition in that aspect?
No, it's
No?
not.
No?
But actually, I'm making it really hard on yourself because I actually want to show. You can't see what's causing it. There's one factor still missing. But the point is, you see this striking landscape with all the patterns in there, and it's not very easy to explain. And so we need physicists for that. Let's put it like that. Anyway, another example: the bushlands in Africa. I already explained that. Or if you go to these bushlands, these sorts of bushlands you find on the top of hills. And if you would go to the side of a hills, it changes. It becomes striped. Does it help in explaining it? What would be an important factor here? What runs downhill? Sorry? Yes, water. Water runs downhill. You remember that. So, anyway, actually, I went there myself. This is a picture. You will see it's a very fuzzy picture. That's on purpose, because if you would look at this and then look at that, you would see how much it has degraded in the past years. It's around 1998, 1999 that I went into Africa. And it was really interesting. So this is how it looks like if you're there. And so actually what you see here is rocky hard soil. And it's like concrete. It's really solid. And while under the trees, the soil is much softer. And so it's different. The giraffe were chased in there by people. They're very nice because they give you a feel for the scale of the trees. But they would never live there if humans wouldn't have chased them in there. And so actually, if you want to understand what happens there, you have to look at the properties of the soil. And you see that actually after a rain shower, the soil underneath the vegetation is much more wet than the open soil, which you pointed to yesterday, Veronique. And that's a bit strange, isn't it? I mean, what do plants do in Africa? They consume water. So why do you find more water underneath the plants compared to the open space? Why would that be?
because the soil beneath the plants is more permeable.
Yeah,
the
exactly.
soil-
Exactly. There's lots of things happening. The roots are there, but also there's lots more soil community, which dig in the soil, and they have something to eat there, not in the open space. So the water infiltrates, and so you see there's a sort of a feedback between plants promoting the infiltration of water, and that in turn stimulating the uptake of and the growth of plants. So a positive feedback. That doesn't sound interesting. And well, what that leads to, of course, I'm sort of stepping over this quickly, but positive feedbacks create alternative stable states. You either have a soil which is rock hard, has no holes, no gaps in it, so water doesn't want to infiltrate, or you have plants that actually there's lots of stuff in the roots in the soil, and the water infiltrates very quickly, and you have a lush vegetation, as you can see here. And of course, this explains why you could have this, well, this is a sort of double well potential, which also Nigel talked about yesterday. And so two stable states, and the ball goes in either state or the other state. Well, does this explain already the phenomena that we've seen about the pattern formation? Would you think like, well, we've solved it? Or not? This one, you shake, not. Why not? Yeah. Yeah, so the straight lines, he says. So the spatial configuration is not explained by the feedbacks. It's just the presence of patches would be explained by that. And so that's actually a good point. And an interesting thing, sort of a trick for that came when it started raining. And so I'm from Holland, so if it rains, then it sort of starts drizzling and it's wet the whole day, and that's slowly the water comes down. Not in Africa. If in Africa, I guess it's the same here. If it starts raining, it gets very dark. It gets pitch dark, really dark, and then an enormous amount of water comes down. I was just amazed by the amounts of water that come down. And within, I think, 10 minutes, there was 10 centimeters of water standing on top of the soil. And it started to run away everywhere, to find the lower places, of course. And so, yeah, as you can see here, really very wet and lots of water. And so what happens then, of course, is that in some parts the water infiltrates, in other parts it doesn't. The water that doesn't infiltrate doesn't stay there, it starts to move. So it moves to other places, and that creates a sort of spatial dependency, potentially. And so we thought like, this is interesting. This we can sort of model, isn't it? Something to put into a model. So we thought of, how do you make a model like this? Well, you need plants growing there. And we're ecologists, so we're not physicists. We don't think like, oh, let's take out every little detail. We were more like, no, you need this, and you need this, and you need this, and you need the butterflies and the giraffe, and so on. And so plants, they evaporate water, and they consume the water from the soil. And the soil results from surface water, and surface water results from rainwater. And of course, rainwater and surface water is more mobile. Soil water is also mobile. It's more mobile than the plants, but it moves, and a little less. And the plants themselves also move, but way less. And so we configured this sort of a model, and it looked like this model. And so also we have a rainfall, and we thought like, well, you know, there's still a bit of water infiltrating even when there is no plants. So we need to have a minimal water level. And the minimal water level is sort of W0. You could say it's 10%. 10% of the water infiltrates if you have no plants. So detailed thing. And and of course the consumption of the infiltration of the water gets into the, don't I have labels? Yeah, so the infiltration goes into the soil water. There's losses, there's uptake, Holding 2 functional response. Why Holding 2? Because every ecologist always thinks like, no, you need Holding 2, because if you don't make it bend up, things go strange. And then and we added growth as a sort of a link with the uptake, and we have mortality. We said like mortality of the grazing and the natural mortality. We never ever split it up, but we wanted to have this explicitly as two forms of mortality. And so this is how we constructed the model. And the model works nicely. So you put the model on a grid. I don't have to explain this to you. You guys know how to make a spatial model. And we have boundary conditions. So that's one thing to mention. We had a sort of a spatial grid where we modeled the vegetation, but we didn't want to have a sort of a whole ecosystem. We wanted to have a little middle section of a part of an arid ecosystem. And so we sort of mimicked then the effect of pre-depletion by the plants standing a little higher or a little further away by imposing periodic boundary conditions. And in this situation, which is on a slope, at least I thought, I'm not really sure what sort of configuration we used here. If you were to put it on a slope, you would have periodic boundary conditions in y direction, and the other ones you could, for instance, also take. You can also make them periodic. You can also just make a sort of a wall and prevent the water from spreading. And then I'm actually this. So this might be a bit confusing. We did have periodic boundary conditions, but the model is on flat soil, so there's no directional flow. I just inserted the plot here, which maybe not so clever. Okay, and so then of course, you have a field, you randomly scatter some plants over the whole field with a bit of noise, and this is how it looks like then. So, patterns explained, great, that was nice. So, at this stage, basically, what we knew of was these models of spiral waves. So this is much better. I mean, hey, that's great. Pattern formation. We have to explain the pattern formation. And of course, you can lower the rainfall, higher the rainfall. You get different types of patterns, and you can also put it on a slope and the whole lot. And actually, it matches the photographs that we had from these era. This was before there was Google Earth. So we were very happy to have a few photographs that actually showed these patterns. And well, different types of patterns, wetter areas, holes patterns, labyrinth patterns, different types of patterns. We could actually get photographs to show these. And then we were thinking like, well, that might be a nice paper. Maybe we can send it to like Ecology or American Naturalist, like Impact Factor 5. And then suddenly, whoosh, this paper came in. It was in 1999, and we were working on these models, and we were just flabbergasted. He had made a model, Blaise Klausmeijer, about pattern formation. Did I include a picture of it? I actually don't think so. So this paper came out, and this was much more a physicist approach. It would be nice to sort of show it was from the group of Simon Levin in Princeton. So I think it was dN dt equals some rain input times some water loss rate. So, well, whatever. I'm not sure what parameters he used. And then there is uptake, which was n times what's the? No, sorry. This is not n. This is water, and water times n squared. And there is dN for the vegetation, which is W times n squared minus mortality times m, and then plus actually here no parameter. So that's just Laplacian operator times n plus some factor, let's call it alpha times W and n. I keep on mixing the n's and the w's. So you saw us constructing this elaborate, detailed model with everything in it, and he came with this, hugely reductionistic. And we were like, you can't do it like that. Well, you could. You publish it straight in Science. We hadn't even thought about sending it to Science. So we've got a bit outclassed there. But this model actually shows roughly the same dynamics as you also had in our model. Our model is a bit more accurate, we think, particularly on the biomass distribution, but the essence is captured already by this model. So we got basically our ass kicked by this guy, which was not a very comfortable situation in that. We actually still published it. His model was solely based. I put here, actually it was not like this. It was like beta times the gradient. He made it always on a slope, so we published it. Let's say we can also have that in situations with on flat soil, and so on. So we have still had lots of nice papers on the model. But it's nice to illustrate the difference between a sort of an ecological approach with all the details in there and making a complicated model, and the quick and dirty approach that was taken by Chris Klausmeier, which is from the group of Simon Levin, is a physicist and much more reductionistic. Okay, well, you might think that, I would say these were the first models that came out that actually came with realistic pattern formation in a real ecosystem rather than these funny spirals. And so, which is, well, on the other hand, you might think like, well, this is completely new. Well, not, because well, you guys know this. These sorts of patterns were already made by Alan Turing earlier on. I don't think I have to explain the story of Alan Turing here. Do I? Who does not know? Who wants to hear the story about Alan Turing? Nobody? Great. Push on. Well, he worked in the war and morphogens, morphogen X, morphogen Y interactions. Remember that there's always this activator-inhibitor framework that he uses, yes?
Model that you had a lot of parameters. How do you fix it? Fix the parameters? It's like you measured the size of the clusters. And
Well, no, we actually went to Africa before we actually started doing the model. So we actually didn't have, well, we had some of these feedbacks. We had sort of measured them, although mostly in experimental setting, not as a, we didn't focus then on getting parameters, but more on getting the principles behind that correct. And there's a basis behind that. And most of the parameters, of course, we try to get them from nature, from the literature, but that's was like this. So we would think the units are appropriate, and the parameterization should be like it should be the case, but we have not measured them.
Okay,
Which is actually the case for most ecological models.
Because your pattern can depend highly on your choice, right, of parameters.
Yeah, sure. But we just have a parameter. We think this is a realistic value, and then we just scan the parameter range, like everybody does. So basically, our approach was not any different from any theoretician making models. You look for the principles behind it. An ecologist would want to have, if you want to make a graph and you put units along the axis, your ecological colleagues will immediately say, like, what do you mean 100 grams per square meter? That's like nothing. You need much more biomass there. And so you try to get it approximately right so that it doesn't look unrealistic, but it's not that you parameterize it per se, particularly not in these African systems, because they're not close by. So I will continue with other examples. We did more effort into parameterization. And tomorrow I have examples which are fully parameterized. Okay, well, so then you have this symmetry breaking instability that you can have. I think it was very nicely explained by Erwin, so I don't have to explain that anymore. What is nice is actually showing that, well, I would argue, Turing, of course, was focused on understanding the brain, and he made this model to understand how a brain would develop. And I think, if I'm correct, Veronique, it doesn't work like that for morphogenesis. It's way more complicated. However, for ecosystems, it works brilliant. And so I want to take you across the world and show you examples of systems which were either shown to be a Turing pattern or are suggested to be a Turing pattern. And although some of them are really questionable still. So arid systems, I've explained them already. So this is a photograph, I think, on a bit of a slope. So you get these bushlands with bare soil in between, where there's hardly any water infiltration. So water is a very important resource here. The inhibitor is water. Maybe I can... No! I didn't want that, sorry. Well, I don't think I can stop it. I will just start over. And we'll follow the route. So water here is a sort of inhibitor, but it works differently than the Turing inhibitor because it's like a reversed inhibitor. When you lower the water available, that starts to inhibit the plant growth. Well, we can go to another location if the thing wants to move on. A little further on, so you can see there's all these hilltops, hardly any human presence, although there's a road. And here is an example of on top of a hill where you don't have directional flow and you get these labyrinth patterns. So the different versions, you find them. And now I'll go into the other side of the world, to Siberia. In Siberia, you have peatlands, strongly nutrient limited. And you get these sorts of tree ridges in the peat. The trees, the higher plants start to accumulate in ridges, and they do that by absorbing the phosphates, particularly, but nutrients, into these ridges. And this is another example in Finland. This looks different because it's underwater, but it's the same system as you can imagine. And now we go to a very important part, which is the next part of my story, which of course is in the Netherlands, where we have mussel beds making patterns. That's the next part of my story. And so now we go to the most beautiful ones, and they are not well studied. So if you want to have a good excuse to go snorkeling on the Great Barrier Reef, you say like, no, I want to study pattern formation in the Great Barrier Reef, and I want to do actually some experiments, and you can test that. And we actually don't know what the processes are. What is striking is that you can have like patterns at multiple multiple scales. You have to see these larger stripes. Within that, there are smaller stripes. So patterns within patterns. And if you go to Shark Bay, on the other side of Australia, you have seagrasses. And they have the same phenomena. Colleagues of mine have gone there, didn't solve the problem. So I think we need a physicist. You might think it has to do with wave action, but that's not the case. So now we go back to our first picture at the beginning of the... Now I want to ask you again, what's causing this pattern formation in the trees in the Rocky Mountains? I want to see some fingers again. Yes. Snow. Exactly. So there's a very wide inhibitor there. So you couldn't solve the riddle at the beginning. So what happens is the wind blows and carries the snow. But these trees, they create a vortex. And the snow drops down on the ground. And it can be meters, mountains of snow, meters thick. And in the other areas, less snow drops down. And that restricts actually the size of the growing season. It's only a few months for the year. And if you suddenly have 2 months less growing season, it's very difficult to grow. The trees have the typical Christmas tree shape. So they sort of, the snow moves away from it, and the saplings underneath the trees, so your dispersal aspect might play a role there. The trees protect their own saplings against the effect of snow. And that might explain, and I think there was a guy in Canada, for the United States, who studied that and measured it and so on, and we put that into a review paper, which we published in 2007, then didn't bother about it anymore. Years later, we looked into the literature, and it turns out that this one guy who studied this got into a fight with lots of other people who were studying mountains, and they didn't agree with him. And it turns out maybe that there's lots of reasons why trees can grow in stripes. It can also be just depressions and so on. So it's not mutually exclusive. But it's sort of striking that we sort of light the fire a little bit and run away and let that guy solve the issues. That was a sort of, and we didn't know because it would have been great to contribute to that discussion. But yeah. Anyway, to continue, I want to take you now to the things that I've been doing myself, mussel beds. It's also a sort of a start. Are there any questions about the previous part? Yes. Not, so the trees grow in concentrations because they protect each other against the snow. So that gives a local facilitation effect and there are self-activators. The wind blows the snow over the trees, then it drops down behind the trees. It creates a sort of a local inhibition effect behind the trees until there's a certain distance that this vortex are sort of finished. And then you get the next stripe. And that's, we think, the explanation. And the picture sort of doesn't show that, but suggests it, so to say. Yeah. I hope this is sufficient.
So when you made that model of the vegetation patterns, did you, for example, look at any like sort of quantitative statistics of the shapes, like for example, the distribution of size of areas or like roundness or things like that to compare the model patterns with the observed patterns?
No, I didn't. Also, the simple reason that I was only a postdoc for a year there, and then I moved to this, and I did much more on this. But I actually, so what people have done is they made an algorithm to scan the whole planet for regular patterns emerging out. So they actually in that way looked for all sorts of patterns and patch sizes and see how they depended on conditions. And you have all sorts of types of patterns and so on. Yeah, so I know the studies are there. I wouldn't be able to tell you more specific about it. You have to look them up. But I wasn't involved much anymore. And apart from that, I think the whole study of pattern formation in Africa has basically stopped because any place that has patterns, it's Niger, Burkina Faso, Somalia, you can't go there anymore because of the Islamic insurgents. Australia has also these patterns. That's still one that's you can go to, of course. Yeah.
Maybe to complement the answer, there is so this is a regular patterns, but there is also irregular patterns, and there people have characterized the distribution of patch sizes and fit those, use other power laws, and then try to fit the exponent. So I can send you references if you want, but basically they track patch size, and then they compute the distribution and try to see how close that is to a critical transition and things like that.
Yeah, that's
Sonia Kefi's paper in Nature.
Yeah, that's true. That's true. Good point that you mentioned that. Yeah, yeah. Although we can have lots of discussions about precisely what she measured, but that's but definitely she touched upon the project, on the problem. That's true. Yeah. Okay. Yes, there's somebody on the other side. Watch out for your computer.
These patterns are often related to fractal formation. So, is that correlations are related to like manage their research because fractal distributions often cover better the space and can manage better their research when they are limited?
Well, a fractal distribution basically has patterns that are repeating at any various ranges of scales. Depends on whether you see them as a fractal model or as a measurement of a fractal distribution. And so in essence, could be, well, at many scale, even scale-free. But if you look at this, do you think that this is a scale-free pattern?
Yeah.
Yes, you think so?
Yes.
This is definitely not a scale-free pattern. This is, Turing patterns create a pattern on a specific scale. And so they are not scale, and then there are even systems that have multiple patterns at multiple scales, and you can see that if you make a distribution of this, various peaks at specific wavelengths. However, if you start perturbing these ecosystems, then they go much more to a fractal shape. But that's not intrinsic to the ecosystem. That's external process. Although that's not necessarily true. You can have particular dynamics in the ecosystem that create fractal distributions. Although I must say, very often they all come down to the same model. Although I shouldn't be doing that, but we all have Turing models. It's the same sort of phenomena. They have a certain particular model that creates these scale-free distributions of patches. Many examples all come down to the same model. And then they have distributions which sometimes are really convincing. Forest fire distributions, for instance, are really convincing, I think myself. And some of them, they think like, well, this is curved. And they draw a straight line through it and say, scale-free distribution. And so I think it's an interesting question whether really how many ecosystems create scale-free distributions out of themselves. And I've also done studies myself where we started with a system with a regular pattern, and we waited at the moment that the winds would start blowing, as you can see in the marine systems, and destroy the patterns, and then they were they approximated scale-free distributions. So in that case, the scale-free distribution is a sort of an indication of approaching maybe a tipping point. And so there's various approaches there within ecosystems. And I think the verdict isn't out there.
Okay, so it's not so regular. There's some range there. It's more found than another no.
So Turing patterns have a specific scale. There are other types of models based on cellular automata, based on the Ising model, derivatives of that in the various forms create scale-free patterns. If you mess up a Turing system with a beautiful regular pattern, perturb it by wind effects and so on, it can also show scale-free patterns. And so that's basically where we are now, I would say.
Okay, thank you.
Okay, so I want to take you to my favorite ecosystem, mussel beds. And that's also, in estuarine systems there are many forms of patterns. You have mussel beds. So basically after a year of walking around in Africa, I got a nice tenure track position at the Marine Institute, where I'm now, and studying marine ecosystems with patterns much closer to home, which is convenient if you're actually more interested in models than in doing experiments in faraway countries. And so they showed me all sorts of examples of patterns, like this photograph of mussel beds, as you can see here. But there's also examples of tidal flats with biofilms of diatoms. And because of the interaction with the water, that also forms a sort of a regular pattern. Another example is this photograph. These are barnacles. I guess you guys don't know what barnacles are, small little animals. And this was brilliantly studied actually in the 80s, explained as an interaction between positive feedbacks and negative feedbacks, while the whole concept of activated inhibitors and self-organization didn't exist even. So, but was very nicely studied by Mark Burtis' group in Rhode Island. And so, well, how do you explain these sorts of patterns then? And that was something that was my question when I started at this Marine Institute. And of course, in the back of my mind, I had these stories about African systems that I worked on. And the question was, can you transport that into the marine ecosystems in the Netherlands? And is that possible? Well, of course, you need to know a little bit more about mussel beds. This is how it looks like when you're on them. Of course, you see the patchiness, but you don't see the pattern so much. But for that, you have to look at higher up, and then suddenly you see that this mussel bed, there's of course two striking things. It's a very concentrated patch of mussels. And apart from that, you have the sort of zebra stripes in this ecosystem. I do always show you my nicest example. This is the most beautiful mussel bed I've ever seen. More realistic would be this one. So what you have is certain zones where the density really became high and they become patterns, and then there are fluffy areas around it where it's not so clear. On the other side, there's also a phenomena of that you see here. There's here a stripe, and then there's here a stripe, and there's here a stripe, and then within that there are other stripes. So you have stripes within stripes, and that's there's much more to mussel beds than simple straight lines. But on the other hand, let's start with the simple straight lines. How do you explain these? And so for that you need to think about. Peter Herman was my supervisor then, and I said like, well, activator, inhibitor, you need positive feedbacks and negative feedbacks. And we started thinking about, okay, what's the ecology of mussels? Well, they are filter feeders, and they consume algae. And the algae sit in the water and flow around, flow over the mussel bed. And so what happens here is that you have the water comes in, goes in, and then goes out on the other end, and again is consumed by the next mussel. So you can see that they are competing for the same algae in the water. And that's something very measurable. If you put a measuring tube just in front of the mussel bed and on the other side, you can see the differences in algae concentration, particularly very close to the bottom. That's, so you see the filtering effects. But that's not all. Mussels are also living on tidal flat, and tidal flats get lots of wave action, and there's lots of birds and a lot. And so to protect against that, they make these wires, as you can see here. And this is a photograph from my lab where there's a rock on the surface, where you can very clearly see the wires that's attached to the rock. But if you're on a tidal flat, there's no rock. There's only one thing to attach to, is other mussels. So they attach to each other to basically protect against these guys, so wave action and predators. And so they form mats. So you can imagine that this guy here, he's fair game. The first bird comes around, picks him up, and he's gone. If you're in the mat here, if you're in the bunch of mussels that you see here, you're actually pretty safe. For a human, you have to put some pressure on it to dislodge them. For the birds, that's not possible. They can't dislodge these mussels from these mats, so to say. So there's a sort of a two-scale effect here. On the one hand, you have competition between mussels for the algae, and the algae flow, and the effect is measurable for meters distance. On the other hand, they facilitate, they cooperate I want to emphasize that to the one who I talked to at the coffee break. They cooperate with each other, and they benefit from each other's presence because they can put a wire to the other guy, and that makes you more safe, and the other guy also more safe. So it's not a classic cooperation, but so a facilitation between the muscles, and we call that skill-dependent feedback. We found sort of a term that we thought like activate inhibitor is a bit fuzzy for an ecologist. We call it skill-dependent feedback. And then we made a model of that, and it takes in consideration that basically you have a separation over the water column between a layer of water that goes over the muscles, in between the muscles, and a higher water layer that sits on top. They are a bit dislodged because of the friction of the water. So you have basically a two-layer system in the water column, and the muscles consume the algae from that lower water body. That becomes muscles. The muscles self-facilitate, lower each other's mortality. There's of course an upper water layer that can replenish the lower water layer. And of course the water flows, and the muscles, they move a little bit. I will show you tomorrow lots of examples of moving muscles. So they move a little bit, but they're still animals, and they don't go as fast as the water. So there's a difference in this flow rate and the movement rate between these two. And so, yeah, any questions about this? Yes?
So the muscle beds, you could say that they form this pattern because of a carrying capacity of the algae?
Let's say that competition for, yeah, you can say there's a certain carrying capacity for muscles in the system, yes. Yes. And, but you also noticed now in the model that we didn't put any carrying capacity in. We have an explicit resource, which is the algae. The algae flow in. Then they are, there's an exchange between the higher water layer in the upper part. The uptake here, we now took a linear relation. Didn't make the mistake again, of course. There's flow of this model, and here also is growth. Again, a linear equation, the simplest possible interaction. Basically, we didn't want to make this more complicated than needed. And then this term here, this is actually important. So there's a mortality term. dm is the specific mortality rate. You multiply it by the amount of muscles to get the population mortality. But the mortality goes down by means of this term here. It's a reversed Hill function that lowers the mortality rate as the m increases. And this is the essential element that differentiates this model from other models like this. And of course, well, you understand that we use a gradient operator for flow and Laplacian operator to model dispersal, diffusive dispersal. So there's a diffusion approximation, as we say. Muscles are way more complicated than randomly moving, but that's good enough for now. So this is how the model then looks like. And if you then have a starting field and you let the model run, it does something like this. It creates nice patterns. And if you make sure that the colors look just like a real muscle bed, then it actually looks pretty realistic, doesn't it? Who believes this? Who believes this? 1, 2, 3, 4. You're dumb. Never believe in ecology. Never believe a modeler. So that's actually, this looks the same. It's a hypothesis. And we actually, I'm not going to go into the testing because that becomes really deep side ecology, but we measured, you can't test whether this model is correct because it's a whole muscle bed. You can't say like, well, I want a muscle bed here with a D that's higher and a muscle bed here that's a D with lower, and then hopefully the muscles will, you can't do it. What you can do is you can look at the assumptions. I mean, is there depletion for algae? Is the mortality really higher when you are outside of the muscle bed? So we sort of tested all these assumptions and put it into the model, and the model, we sort of got parameter values for the logical things. But of course, if you want to look at, like, we have a C here, a consumption rate, the rate of consumption of the muscle, of the algae by the muscles. Yeah, how do you measure a C? It doesn't exist because this is a very simplistic relation. So you just parameterize it so that it looks actually like what you see in the real world. So that means also that you adjust your model to look like the real world. So it's a hypothesis, it's not the real world. Yeah, so what I can show you, and that's maybe nice, is what happens actually in the real world. Just a series of photographs. So this is a photograph of a mussel bed taken by that man there, Norbert Dankers. He knows a lot about mussels. Near his house, a mussel bed dropped down in 2009. And you see here this whole scattering of small little mussels over the mussel bed. And what you also see is that there's sort of a gradient here. And the sea is there, so the food comes from the water. And there's much more mussels here on the edge than there is over here. And you might think like all these little dots that are mussels. Well, no, they're not mussels. They're clumps of mussels. The mussels here in this picture are, this is a €2 coin. Well, it's about this size of a coin. And you can see that the clump is just the size of a coin, and it contains 25, 50 mussels. So this is how it starts. And so it's still already very patchy on a very small scale, but on a larger scale, it's quite homogeneous. Well, if you then come back a little later, you find actually it has become a little patchy. And it's not really striped now yet, but what we think here is that actually there has been some wave action, and they're just eroding mussels then. So they just roll towards each other and get stuck to each other, and you get a sort of a separation between patches of mussels and patches without mussels. And then if you wait a little while, you see that actually the mussels start to build little hills, and that basically they eat anything in the water. Anything that sits in the water comes in them. Well, what's in the water? Well, algae and mud. The mud, they have a way of separating that, and they excrete that in the form of feces, pseudo-feces, and that drops down because it's a pellet. And so and they crawl on top of that. And so what you see is that slowly little hills of mussels start to occur, which actually is a beneficial effect because you push yourself up through the lower boundary layer into the higher ones where there's more food. So there's a feedback there that we didn't put into the model, but which does exist. So you see the real world's actually a little bit more complicated than our model. And after a little while, it starts to look beautifully like this. Don't look at the Dutch mountains in the back. So this is a different location. This is in Wales. In Wales, they have the most beautiful mussel beds in the Menai Strait, which have mountains on both sides. And that prevents it from the waves from destroying a mussel bed. That one that I showed you was in the winter was a big storm and it was smashed to pieces. There was not much left of it. That's the real. Only 10% of every existing mussel bed survives the first winter. So that's why the article is called "Pattern Formation in Young Mussel Beds." Old mussel beds only look so beautiful here in this place. It's really great. Okay, nice, patterns and ecosystem function. Let me have a look what I'm going to talk about now. And so, what is the effect of patterns on ecosystem functioning? That's an interesting question, isn't it? So what can we expect here? What do we think? Is this important, or is it just pretty patterns? Of course, we had this paper in our mind in 1997 who said like, pretty patterns, it's only for looking, it's not really important, doesn't really exist actually. We showed it exists. But is it important? That's the next question. So who thinks that this is important with an argument behind it? Yep, you were first.
If you take the example from the desert, the pattern facilitates the intake of water. So
yes, so you have an effect on the ecosystem by the formation of the patches of water, and that's patches of vegetation because they reserve and they store the water actually. So that's one thing, and with the mussels. Sorry? Yeah. So mortality is of course lowered if you start to aggregate all your cells to the same. Yes.
For the case of the species that does bioengineering, it can change how other animals behave in that area. No.
Yeah, I think the little mountain building is a form of engineering.
Mm-hmm.
Yeah, but I would also want to return to your suggestion of a carrying capacity. So how does a carrying capacity come into play here?
It can increase or decrease the carrying capacity because they change the...
No, the amount of food is determined by the algae, and the amount of algae coming in is the same. But it's basically, you have a certain carrying capacity. It doesn't fill the whole area. You can't fill the whole area with mussels. The carrying capacity is too low for that. Of course, you can just lower overall, on average, the amount of muscles, but then they face a high mortality rate. So what's the best way of dealing with that? Is to aggregate into patches. But then, and that if you then align yourself perpendicular to this, to the flow, you become a sort of an algal harvesting system, and prevents high mortality rates. Any other ideas?
You showed that you, like for instance, in the arid ecosystems, if you tune the amount of water, you get different patterns. So if you observe the large-scale patterns, could you determine how that, like the health of the environment, essentially?
So,
for instance, if the amount of so basically, if the large-scale pattern changes a lot over time.
Yeah, sure. That's a very good suggestion. We had a paper, basically just with that idea, a paper in 2004 in Science, simply saying if a pattern changes, conditions are changing. So that's a very good idea. Yeah, you are only 20 years too late. You're 20 years too late. Okay, so yeah, increased production because of water use efficiency or net production or a higher rate of recovery after disturbance. Would that be the case? That was an interesting question. Is there a benefit in terms of its resilience? That would be a possible thing. Or that's the ability. I mean, is there maybe, you can say there's a rate of recovery, but maybe you never recover. And so does it make the pattern survive in conditions which would otherwise not be able to survive? Or can you resist change with that? So increased resistance. So all these questions are basically there. And so we did that, we started studying that, and we do that in the way that an ecologist does that. You sit behind a computer, you press run. And so what we did is we made a simulation, and we thought like, well, we have two situations. We reduced the amount of biomass by 2/3, gone muscles. And we have two situations. One situation is we leave the pattern intact. The other situation is we reshuffle the pattern. So every cell keeps its own biomass, but we just mix it all around a moment. And what happens then? Well, you can see here, if you leave the patterns intact, it very quickly recovers to the old density in the model. If you don't do that, if you homogenize the patterns, it first collapses even further, because the loss of the pattern imposes a lot of mortality. And in this period here, you see that the pattern reestablishes, and then you go back to the same situation with the developing pattern. You had a remark, wasn't it? Or somebody who was pointing his finger. Okay, and so, well, that was one element that we took out as an effect of the pattern formation. Another one is a bit more complicated, is we wanted to study how this pattern changes. It actually comes straight along with your suggestion. What happens if you have changed the parameters of your model? If you have lower rainfall in arid systems, or if you have increased mortality because climate change and more wave action and therefore more muscle mortality. How do you study that? Well, that you do what's called bifurcation analysis. And so that was, nowadays there's specialized software for that called Auto or Content. It's not easy to use, but you can use it, and there's a very thorough analysis. Didn't exist in 2005, particularly for advective systems. You need to have a stable system for that. But the essence of it is that you take a small little section of your system and you impose it with a single peak. You say one peak fits in there. And then you basically, using continuation software, you follow that equilibrium, changing the patterns, using some algorithm that actually traces that equilibrium. It can trace it through stable regions. It can also trace it through unstable regions, which you can't do with normal simulations. But in 2005, I did it just hands-on. I made a huge program that computed it all. And it looks like this. And so what you see here, I'm going to describe what I see, and I'll leave you to draw the conclusions. So here is the homogeneous equilibrium. And it goes down steeply to a certain point where it hits the bottom and no muscles can survive. Through this striped region, this model gets Turing unstable, and the patterns, a small little disturbance will give you the patterns. If you then have pattern formation, then you see here the red line is the peak of the pattern, much higher than the average. The peak of the pattern, and it forms a little a curve down, and you have an unstable part here, and you have here the average pattern. And so, of course, see that with this system, it can all go all the way here. The average biomass of the... Oh, yeah, sorry, that's not clear. It's the biomass of the average biomass of the pattern state. Good that you remind me to do that. And so, okay, what can we learn from this graph?
Well, you can see that if you reduce the amount of available food, I guess.
Yeah, I didn't explain that, sorry. That's along the x-axis, is indeed, thank you for reminding me that, is the amount of food. And you look at the A up, the nu is basically a non-dimensionalized version of the model.
After the pattern formation, you have a higher average of biomass for a lower content of food than the homogeneous equilibrium.
Exactly. Does everybody get that idea? So if you compare this with that, you see that biomass is much higher. Here it's just a little effect. At high algal supply, it's just a small effect, but it gets bigger and bigger and bigger proportionally as the amount of algae is decreasing. The algal input rates.
Where you have like a lower value of biomass and you reduce the available food, and it shoots up when you have the fat.
Yeah.
Right? And they all transition between the homogeneous and
Yeah, so you see that the first shift towards a pattern state is already giving a bit of a boost in the alcohol, and the resolution is here not good enough to see it in more detail, but it basically curves down slowly. Yeah, but there's more. That's actually the most important part. Anybody? So how many mussels can survive here if you have no pattern? No mussels. And if you have a pattern. So the mussels can survive under conditions with patterns, which they cannot survive when they have no patterns. So it actually broadens the range at which they can occur because of the pattern formation. And they can survive with lower amounts of algae until there's only one drawback. So if you look at the mean, sorry, the peak biomass, you go down, 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 and you still have a bit of mussel bed left, and then you're over the threshold, and boom, you collapse down. So there's a tipping point, because you can survive in a pattern, but if the pattern gets less and less and less, at some point it's not big enough, and the whole mussel bed collapses. And so you go from a continuous system that gradually goes down to zero to a discontinuous system, which has a tipping point. Okay.
Sorry, Johan, maybe I have a question. How does the wavelength of the pattern change as the environmental conditions worsen? Do you see a change in the characteristic scale? Or?
You're going too fast.
Okay, sorry.
Too fast. So that's one thing. Thank you for sort of pointing to the thing. So I want to draw this line here. I'm going to do it roughly. So here we have A up. And here we have our, well, let's say the homogeneous equilibrium. And here we have the patterned equilibrium. But this is for a certain small little peak. We only work with a small little peak. There's a very small world here. And we made this a certain size. So what if we just make this size a little bigger? Then you have, well, sorry, you make the size bigger. What can then happen is, well, sorry, so you make the size bigger. What changes then? What changes if you have to make the size a little bigger? If you see this as a little section of a very big world, then of course this goes to there's the next one, here's the next one. But if you take a space like this, then here's the next one, and here's the next one. So what's the difference between these two? It has a different wavelength. So who sets the wavelength? So we artificially set the wavelength here. And of course, you can have other wavelengths. So let's take another wavelength. So I'm actually not sure. This one's probably a bit lower. So what you can have then is that maybe it does like, it goes like underneath here, and then it goes. So is this important? Why is the blue one different than the white one? Except for having a different wavelength. Of course, you spread it out over a bigger space, so it gets a little lower. But also, it can survive for longer. So which wavelength is the system in? Do you know? How do we know? We set the wavelength. The system can have all sorts of wavelengths, isn't it? And you can make different ones. You can also look here, and there's also one here, and there's one here, and so on. So suddenly, we don't have this one line anymore. We have a whole cloud of lines emerging here. And so the world is a bit more complicated than this one line thing that we did in 2007. And so actually, what you can do is you can make a plot where here you have the food availability, and here you put the wavelength, something like this. And so you have like this wavelength being stable here, this wavelength being stable there, this wavelength being stable here, and this wavelength here. And so meaning that this is a very short wavelength, isn't it? Am I doing this properly? This is or is it the wave number? Sorry, wave number. So the amount of waves that fit in a certain space. I would explain. You have a certain space, and you allow either one wavelength in, two wavelengths, three wavelengths, four wavelengths. So what you then get is that here you have, this is 1, this is 2, this is 3, this is 4, this is 5, this is 6. And you're changing the system. So what happens then? You're here. And so what happens then, you think? So the alkyl density goes down. So you start here with certain wavelengths. You have 6 little waves in your field, and you move that way. So what would happen? You drop down, 5 wavelengths. You go further, and you go 4 wavelengths. Actually, you need to be a bit slow, though. If you do this very fast, what will happen is that you actually move like, you go further, and then it's up. Only here the system gets the idea that actually the wavelength is wrong, and it goes immediately to 4. So the speed actually is important. And so what is actually important here, there's lots of details about how this exactly works. I don't want to go into too much attention with that, but basically what this means is that we don't have a single line. We have a whole space here which has all sorts of possible stable wavelengths. Loads of them. And what is the consequence of this? Has huge consequences for pattern systems. Yes?
The robustness to perturbations?
Yes, but that's very broadly. But so is the robustness bigger or smaller?
You mean as opposed to if there's a single line?
Mm-hmm.
It's bigger if there's an area because you can sort of move through the phase space without.
Yeah. But if you can even draw it further. Don't forget that if you only take a single wavelength, you get this big thing, and it goes to here. And then basically, you remember that I explained that if you go along with a single wavelength, you have a tipping point here, and you drop down. System gone. But if you have this whole envelope, you don't have the phenomena anymore. There's no tipping points anymore. So that's an interesting thing. So actually, do you guys know the name of Martin Scheffer in ecology? Martin Scheffer in ecology is a name. He works on tipping points. And so basically, ecosystems have certain points where, because of conditions worsening, you go from one state, and you fall to a different state, a degraded state often, which is a very important point. And actually, this agrees with this idea here that you have you follow this line and you drop down in the collapse of an ecosystem. It's actually is also explained already to by urban in a slightly different context. But if you have all these patterns, you don't have it anymore. And so that's actually something that we recently worked on. So basically, this envelope is called the Busse balloon. It's just simply a guy called Busse, and he liked balloons. I find the whole balloon a horrible term, but okay, he calls it the Busse balloon. It's this envelope of all sorts of possible configuration of these systems, independent of its wavelengths that it has, with all these possible wavelengths. And so, without the Busse balloon, a system with patterns, or let's say even without pattern, has this tipping point because of this potential positive feedbacks. But with the Busse balloon, this whole phenomenon is gone. And that's a very important topic, statement, because, well, where do we find all these self-organizing patterns in ecosystems? Where there's hardly any humans. In unspoiled, pristine ecosystems. They have all these patterns. And what are the best examples for tipping points in ecosystems, what Martin Scheffer is working on? They're for shallow lakes. It's a bit of a Dutch example. They are human dugout lakes. They have about a depth of 5 meters. They are very square-ish, I mean, in depth, because they're dug from peat digging. So they are highly human-modified systems. And that's the best example of tipping points, is in this highly human-organized ecosystem. While in these natural ecosystems, there aren't that many possibilities for tipping points, if you have self-organizing patterns in them, of course. Let me put it differently. These self-organized patterns are a sort of a capturing thing for these tipping points. They can prevent tipping points, given that it doesn't go too fast, and that you have not lots of human activities messing up the patterns, of course. If it goes faster, that's a bit of a worry with climate change. You can say, oh, climate change is saved. We are saved against climate change in natural ecosystems because of the patterns. Yeah, if it goes slow enough. Otherwise, you just overwhelm the system and it still collapses. You need to give it time to change. And so this is what we basically, this is the initial idea, is what you mentioned. It was a very good idea. We published that in 2004, and we said, like, well, there's a tipping point in the system with a certain wavelength, and you have a changing pattern that indicates when you have a tipping point. And that's what we thought in 2004. And in 2021, we thought, well, it goes a little further. You actually, with all these patterns, you don't even have a tipping point anymore. Or even, let's put it differently, the whole phenomenon of tipping points becomes much more complicated. And that's what we published in Science in 2001. And of course, you get all these different wavelengths, meaning that with the same Turing system, you have all sorts of possible configurations. But then we sort of thought, like, well, we got enthusiastic and started to fantasize. And we thought, like, well, yeah, there are actually all sorts of patterns. You have systems with Turing patterns. You have systems that have Turing patterns within other patterns, so multiple patterns, complex patterns, which is basically these pictures. But there's also what Max calls these coexistence states, is basically, well, it's a more generalized term relating to systems with two phases. So a phase separation system would be considered as a coexistence state. I think that's still open, but it's an interesting thing to investigate how a system with these phase separation dynamics differs from one without phase separation dynamics. Okay, yeah, with that I would like to finish. So I have a nice collection of pictures of all sorts of patterns in the world. I hope I have convinced them that they're really important, could be really important. There's loads of nice examples from incredibly well-studied systems like here to the non-studied coral reefs, or the coral reefs are not in here, and peatlands have also not get that much attention. So there's lots of stuff to do still here, and they're often studied also by ecologists. You've seen now how lazy models they make much too much parameters, much too much detail, so you can do much better, isn't it? So I would say, look at it. Okay, and to come back, we had skill-dependent feedback, which is the relabeling of Turing's activator-inhibitor system, but then sort of put this in an ecological context, and we pointed at the emergent properties of the pattern formation. Okay, this is the end of my talk. Yeah, so, well, can I rephrase that? Is the question: is a pattern always beneficial to the functioning of an ecosystem? Is that what you mean, or is it something different? Yeah, well, but I would actually don't know. I wouldn't know that, and that's because I think in principle it shouldn't be. It doesn't have to be, and that's I think a really interesting thing to look at. On the other hand, if you just think about what configuration is needed to have these patterns, well, you need organisms that facilitate each other, help each other at close distance in whatever way they can do that, and that any negative effect is put at distance. And that's, of course, a very generalistic thing. You can easily imagine that evolutionary process just would facilitate that, would go for these sorts of configurations. So that's a possibility, not anything that I have any clear-cut, either theoretical or empirical evidence for. But I think that's really interesting is we see these configurations everywhere in ecosystems that have these local facilitative interactions and long-range negative interactions. Is it something that sort of comes out of the processes that drive life? I don't know. And it's very common. If you want to, of course, nowadays we have all these ecosystems, but for 3 billion years it was just stuff sitting on biofilms. And if you look at the geological record, if you go beyond for what's left of something that's for longer than Precambrian landscapes, they're everywhere patterned. They're these stromatolites, all these shapes, and some of them are true stromatolites, other are buried sedimentary structures, and they're all like wibbly and wobbly, and it's simply, I think, because the same setup is repeated also there. And you can actually still, we have a study that actually argues that we can still see these sorts of ecosystems from the Precambrian in our current estuaries, because these biofilms, you saw them here. It still works. And so this is, I think, a very nice topic for future research. I'm not an evolutionary biologist, and it's a hugely complicated topic. And so that's sort of the edge of where I've come. But I think it's a really interesting question. Does Turing's principle come out from evolutionary principles, processes in nature? Yeah, sure. Yeah. Yeah.
So far you have explained your patterns using growth and death models, right? But I know that you have a PNAS that explains muscle patterns using movement, density-dependent movement, without growth and death, right? So what's the difference there? Is another scale of patterns or something like that?
It's just a different scale of patterns, but not necessarily. I think that's my talk tomorrow.
Okay.
And so I can explain, but yeah.
So a question, but also relating to the previous one. So I know there are some groups that, when things have desertified, they try to introduce vegetation again, but then in a smarter way, right? So instead of putting all...
And that works brilliantly.
Yeah.
Yeah.
So maybe...
Digging these holes and mimicking the patterns.
Yeah. And so basically models like this could help you think, okay, for these densities, what would be the preferred packing possibly that might facilitate their outcome?
Yeah, of course.
So you could envision to use this as a predictor of, like, what spacing should you use, or what's the...
Yes, modelers could do that, but I always say, why take a model if you can just observe? And I think there's plenty of situations where we can observe the conditions and the patterning. It would be very nice for an AI system where you correlate these with each other.
Yeah.
And you can then predict what's the best wavelength, basically using, without a mechanistic reason for that. Because you can just look at similar conditions in situations where the patterns are still existing. And that gives you the right wavelength.
Yeah. And so in the same light, because lots of, there's a lot of climate change, lots of habitats are changing, right? Is it possible maybe to not just quantify the amount of species, but the pattern in which they are, as a diagnosis almost to the health of that vegetation? Do you think we could go that far, or?
Well, we already took that up as an example where we looked at how what's this picture? So you go,
There, yeah.
there's a predictable change in the patterning as you
Indeed.
go for either with lower water amounts or lower algal amounts, or if you reverse the whole plot, then you say higher stresses and so on. That gives you, that just flips the thing around. And so you could use that as an indicator.
Yep.
And there's actually also other examples where this is, Turing patterns are brilliantly regular.
Yeah.
If you have a sort of a heterogeneous disturbance, you go from a regular system to a less and less and less and less regular system, and then it disappears. So that sort of indicates that
The tipping point is getting
the
there.
tipping point is indicated by the regularity of the system. And so there's all sorts of ways to sort of indicate this. Of course, you talk about the health of the ecosystem.
Or to know where it is on that scale, right? So
Yeah.
if you're moving, so you can sort of predict. I mean, I've never heard about these like spatial images actually being used to try to infer a bit.
You can point to change, but it's well possible that under certain conditions, for a specific situation, this ecosystem, this pattern is just what you need. While on other situations where actually there's more water and
it's case specific,
and then,
definitely.
and then you actually, this is good and this is actually going bad. So it depends also on what your expectation is, of course. And so, of course, health is a bit of an, we have this human, which is a highly controlled ecosystem. You can sort of say like, well, you know, this is how it looks like when it's good, and if it looks worse, then it's not healthy anymore. But these are not, ecosystems look different under different conditions. That does not mean that they're not healthy. Maybe that's what you can expect during these conditions. Yeah.
More questions?
Okay.
So maybe just to complement what Veronica just said about this, creating the patterns, there are many farming populations in Africa that do that too. So I mean, maybe we are not, yeah, so maybe we are not inventing anything with these patterns. They create holes regularly distributed to optimize the use of water.
Yeah, there are there's a specific number of NGOs are active in that field, and they have the most beautiful websites explaining how this works, and
it's really cool.
Hoping you to contribute. Yeah. And so yeah, if you want to see how this looks like, there's plenty of examples of, and that's actually it works, but simply by making a gap, making a hole, that also stimulates infiltration, and then you put plants in it. And that's, I haven't seen any studies that look at how that looks like 30 years later. That would be interesting to study in 30 years, I would say. Yeah.
Great, so let's thank Johan again for a great talk. So we come back at 2.